All right, so let's get started with our first lecture then on nucleic acids. And in this lecture, we're going to be learning about the structure and biochemical properties of nucleic acid polymers and why the structure and biochemical properties of nucleic acids has made them nature's molecule of choice for genetic information storage. So, I mean, you could imagine that cells could have chosen proteins or lipids or something else for this function. What is it about nucleic acids that makes them so well suited for the role of genetic storage and replication and transmission? And once you understand the structure and biochemical properties of nucleic acids, the reason for their selection for genetic information will be very apparent, and it's actually a very elegant scientific observation. Now, before we dive in, I'm just going to say that at the start of each lecture, I'm going to put a number of learning objectives up on the slides. And essentially, if you can understand all the material that's on these slides, you're in excellent position for any questions I'm going to ask on the exams. So this is the core knowledge that I feel like you should be paying attention to and listening for as we go through the lectures. And similarly, I'm going to list off a number of things that I think you should be able to do. And again, as long as you can do everything that's on these objective slides, then you're going to be in really great shape to be able to answer problem sets and do exam questions that I might ask you about. So as you're listening to the lectures, kind of pay attention for the different things that are on the objective slides, and at the end of the lecture, make sure that you know everything that you need to for that purpose. And if something's unclear, you can go back and watch the videos, and if it's still unclear, you guys can ping me and make sure that if there's some topic that I told you that I expected you to be able to understand, that I presented you with the information needed to understand it. So hopefully this will provide a kind of roadmap for you guys to be able to know how to listen to the lectures and know what to take away from them. Okay, so let's get started then learning about nucleic acids. So what is a nucleic acid? Well, nucleic acid is a polymer built up from nucleotide monomers. And so just in the same way you learned that a protein is a polymer built up of amino acids, nucleic acids are polymers built from nucleotides. And so nucleotides are the building blocks, and the nucleic acid is the polymer. And so what exactly are nucleotides? If these are the building blocks, then it's important that we understand the fundamental monomeric unit of this polymer very well. And so nucleotides are molecules that are composed of a base, a sugar, and a phosphate. And I think the core thing to ground your thinking about a nucleotide is the sugar. So that's this component right here. It's a 5-carbon sugar, also known as a pentose. And we can number, the standard numbering for the sugar is that this is the 1', 2', 3', and 4', and 5' positions of the sugar. And having that numbering in mind makes it easy to understand the overall nucleotide structure, because the overall nucleotide structure basically involves substitutions of the sugar with different components. So for example, the base of a nucleotide is connected to the 1' position on the sugar. So the base goes in the 1' position, and the phosphate of the nucleotide is attached to the 5' position of the sugar. And so from a starting sugar alone, by simply attaching a base to the 1' position and a phosphate to the 5' position, we now have kind of a picture of what a nucleotide is. Now, it's important to recognize that the nucleotides of RNA and DNA are different, and the way in which they are different is in the 2' position of the sugar. So in RNA, the 2' position contains a hydroxyl group. That would be standard for the sugar. But in DNA, this hydroxyl group has been replaced by hydrogen. And so that is actually the defining difference between RNA and DNA nucleotides, is what's going on at this 2' position. And then finally, as we'll learn soon, the 3' hydroxyl position of the sugar is going to be really important for a lot of the chemistry that goes on in building nucleic acid polymers. And so really put a picture of this nucleotide, burn it into your memory, because we're going to be talking about it a lot. So let's talk a little bit more now about what these purine and pyrimidine bases are. And so these are what are called the nitrogenous bases of the nucleotides. And there are two types: purines, which are these large 9-carbon or 9-membered rings like you see here. And there are pyrimidines, which are these 6-membered rings here. And we call them nitrogenous bases because they're not carbons at all positions. There are nitrogen's atoms inside the ring. And again, this is one of those things where you really want to know what a purine and what a pyrimidine looks like. And some helpful mnemonics that I've used for a long time is that purine is the shorter word, but it's the bigger structure, and pyrimidines are the longer words, but the smaller structure. And so that's a mnemonic I've used since undergrad to help me remember which structure is associated with which word. And as we'll learn, the ability of different pyrimidine and purine bases to interact with one another in what are called base pairs is going to form the basis of molecular information in nucleic acid polymers. Now, within purines and pyrimidines, those are classes of nucleobases of which there are specific molecules that are involved in nucleic acid structure. And so within the purines, there are two purines, adenine, which you see here, and guanine, which you see here. And these are used in both RNA and DNA. And then within the pyrimidines here, there are three types of pyrimidines: cytosine, which is used by both DNA and RNA; thymine, which is used by DNA alone; and uracil, which is used by RNA alone. And it may seem a little annoying, but you'll really need to know the structure of these bases. And that's because the structure of these bases and the substitutions to the purine or pyrimidine scaffold is going to be really important for dictating how different nucleotides can interact with one another. And so it's not just memorization. The structures are going to be really helpful to understand many important aspects of nucleic acid biochemistry and biology. And so I've made a companion video walking with you that will kind of walk you through how I've learned to internalize these bases in my mind over the year. And I think if you go through it with me, it won't be as annoying or as complicated as it seems. Okay, so the base of a nucleotide is connected to the pentose sugar, as I mentioned before. And when you have a base connected to a sugar, you have something which is called a nucleotide. And so for RNA, that sugar is ribose. And for DNA, the sugar is deoxyribose because it lacks the 2' hydroxyl. Now, the 5' and the 3' positions of the sugar are the key positions involved in building the nucleic acid polymer. So the 5' position and the 3' position are really important and critical ones for you to think about when we're talking about nucleic acid biochemistry. And lastly, the presence or absence, I want to say it one more time, the presence or absence of a 2' hydroxyl is the defining difference between RNA and DNA. So you really want to make sure you understand that. So the way in which a nucleobase is connected to the sugar is through, as I mentioned before, it occurs at the 1' position of the sugar, and is a type of linkage called an N-glycosidic linkage. So, or an N-glycosidic bond. And this is a terminology that you're going to want to know, because sometimes we're going to learn about enzymes that cleave glycosidic bonds, and you'll learn about other bonds on this nucleotide structure that can be cleaved. And so you want to get that terminology under your belt here, that this is an N-glycosidic linkage between the sugar and the nucleobase. And then finally, a nucleotide is what you get when you take a nucleoside and you attach a phosphoryl group of some kind to the 5' hydroxyl position through an ester linkage. So now, instead of it being an OH in this position, we have some sort of phosphate moiety that's been attached there. And so there are different examples of nucleotides. You can have nucleoside monophosphates, where there's a single phosphate group in this. And this is kind of what the final nucleic acid polymer is going to be composed of, a bunch of nucleoside monophosphates. But there are other types of nucleotides, for example, nucleoside triphosphates, like ATP, which I'm sure you've heard about. And these serve as high energy building blocks that are used by cells to actually synthesize the nucleic acid polymer. And so from a starting nucleoside, you can create a variety of nucleoside monophosphate, triphosphate, diphosphate nucleotides. And the key thing there to remember is that there's a phosphate group in that 5' position. Okay, so let's remember now that RNA and DNA differ in the structure of their nucleotides and the bases that they use. So again, RNA is ribonucleic acid, and it has a 2' hydroxyl, whereas DNA, or deoxyribonucleotides, have no 2' hydroxyl here. And this may seem like a very subtle distinction, but it's going to end up being really important as we will learn in terms of nucleic acid structure and the biochemistry of these polymers. In addition, the choice of bases, as I mentioned before, is distinct for DNA and RNA. So DNA uses adenine, guanine, thymine, and cytosine for its bases, whereas RNA uses adenine, guanine, uridine, and cytidine for its bases. And so that U versus T is another important distinction of the RNA polymer compared to the DNA polymer. And that's something you're going to want to know as well. Okay, and so at this point, I have this recap slide here, which is mostly about the nucleotide and nucleoside structure. And really, like I said, you're going to have to internalize this information. Pretty much, it's the foundation for understanding most nucleic acid biochemistry and structure. And so we really want to take it away very clearly. And so you want to have this picture of the nucleotide in your mind, the base, the sugar, and the possible phosphate groups. You want to know this numbering: 1', 2', 3', 4', 5'. You want to know that the key difference between DNA and RNA is whether this 2' position is a hydrogen or a hydroxyl. And you want to know that the difference between a nucleoside and a nucleotide is whether or not there's this phosphate at the 5' position. And of course, you want to know all the structures of these purines and pyrimidines. And like I said, it may feel like a lot to memorize. It's not too bad. And I think if you just go through the video with me and you draw stuff out, drawing these things out makes it really easy to internalize this info. And once you've got this information under your belt, you're going to be in really good shape to understand the next video in which we're going to say, how do we take these building blocks and actually put them together into polymers? How do you take nucleotides and make polymers? And what is it about those polymers that defines their structure and how they encode genetic information? And so that's what we'll be getting to in the next video.